our first speaker is Jim Kirkland from Cedars, talking about his Translational Geroscience Network.
You don't have to use this microphone if you are a person who projects your voice, but if not,
I'll start off, and you've probably gone over a lot of this before, but as you're aware, aging processes begin at or before conception. We're always aging, so that's why in ICD-8 they took aging out as a disease classification because a 6-year-old is older than a 4-year-old. The new ICD classification that geriatricians often will be using is in ICD-11, and it's impaired intrinsic capacity, but that's not a CPT code yet. But at any rate, things like Down syndrome are driven by acceleration of fundamental aging processes, and these occur in the oocyte before it's even fertilized. PTSD can be a mother can undergo PTSD 10 years before they become pregnant. You find acceleration of fundamental aging processes in their offspring. So way before conception, these fundamental aging processes can begin to operate. And there are many, many things as you've gone through that can influence the rate at which these processes will occur, including a lot of disease states, a lot of things in the exosome, and various kinds of drugs and other exposures. And these fundamental aging processes you can group in anywhere from 4 to 15 categories, pillars, or hallmarks of aging. They all tend to be interrelated in what's coming to be known as a unitary theory, such that if you target one, you tend to affect all of the rest. That's not completely proven. There are some outliers like alpha estradiol that may be a bit of an outlier, but generally they tend to be interlinked. And then the effects of accelerated operation of these fundamental aging processes include many acute diseases as well as chronic diseases throughout the lifespan, from early childhood on. And in elderly people, they include things like the geriatric syndromes. They also include, as in the new ICD code, impaired intrinsic capacity, that is decreased resilience, decreased ability to respond to, say, wounding, to have an antibody response to a vaccination, or to recover after surgery, as well as decreased reproduction, aging phenotypes, and decreased remaining survival. So there are many classes of interventions that appear in preclinical models to have an impact on these processes. Some have an impact only predominantly on health span, others on median or maximum lifespan, some on both. So it's turning out a lot of things in the ITP that were shown to increase maximum lifespan in rodents actually decrease health span. Some of the things that you think about, like caloric restriction on rapalogs, can actually decrease health span. Animals that are calorically restricted living under ideal conditions where they've got constant food, bedding, lighting, temperature. They're the first ones that would be killed by a cat or die if there was a rainstorm or cold exposure. They sleep 20 hours a day. They don't reproduce. They're because they're too inactive to. So don't always. I think the field is beginning to appreciate more and more that health span is not necessarily interlinked with lifespan. And as a clinician, my interest in the patients that I see's interest is in health span. It's not necessarily in lifespan at all costs. So one of the things, I'm not going to go into a lot of the basic science side of these things. Mitzi went over some of it with respect to senescence, and you've probably gone into some detail about the other processes. But I'm going to talk about a little bit more of how do we translate these things through to a clinical application. One of the things that the NIH has established and has got a good enough score to be renewed and theoretically should be renewed, but who knows what's going to happen, given the current situation. It's been operating for 6 years now, been hopefully renewed for another 5. It's a translational geroscience network. And what this is, it's an infrastructure to help people with clinical trials. It doesn't actually fund them. So a lot of the trials involved in the network are funded by the NIH, by foundations like the Alzheimer's Foundation, Alzheimer's Drug Discovery Foundation, some of these, as well as other foundations. Some of the trials are run by private entities, some by individual philanthropists, and some by the military. So there are 4 trials underway by the US military, for example. And what the network does is to assist with study design, IRB language consent forms, preparing INDs, because a lot of these interventions, if they're repurposed drugs or natural products, drug companies tend not to be interested in them because they can't get composition of matter patenting. So that means we as academics have to do this, and it's a tremendous task. I've had to do it with the FDA and with EMA, and EMA was far worse. So if you're using, the minute you're using a natural product for a disease indication, it has to be approved by regulatory authorities. The FDA, for example, it was a 380-page long application. Every page is hard as writing on R1. And 2.5 years. The EMA was 1,200 pages for the same natural product.
Oh my God.
So, and that took about 6 years. And finally, it was granted in December in Denmark. So there are a lot of administrative hurdles in getting some of these things through to actual application. One of the parts of the Translational Geroscience Network is the Facility for Geroscience Analysis. And what we're trying to do, where feasible across trials, is to measure similar things, a floor of things like cognitive function testing, physical testing, wearable devices, at least so there's some commonality across trials, as well as biopsy interpretation where those are available, and liquid biopsies, blood, urine, saliva, and so forth, and storing samples for other kinds of assays, and trying to get consent forms written in such a way that subjects would allow their biospecimens to be used by other investigators for reverse translation. The NIH has allowed these samples, there are about 45,000 a year, to be stored in Frederick, Maryland in the NIH repository, and they're not charging for that. And then what we're trying to do is analyze data across all these trials, irrespective of the intervention, the indication, or the outcome, to look for things that are in common using some new statistical methods, sort of second generation lasso methods and things. So currently most of the trials are in the US at the moment. A similar network was just established in Canada 3 months ago, funded by the Canadian government, which is based on the US network. The EU has just initiated a network. And there are talks about it in other countries. Well, Singapore has a network as well. And the hope is eventually to bring all of these global networks into a room together to begin to get some commonality. Now, one of the things about biomarkers is it's not really reasonable to establish standards, because every few days there's another biomarker that comes out that's better than previous ones. So this will probably be an iterative process when it comes to that. So the trials are for a range of indications, right from what happens in childhood cancer survivors with accelerated aging state they can develop. That trials with St. Jude, looking at senolytics in people who've had chemo or radiation before the age of 10, because they've got over 800 patients, for example, who develop an accelerated aging state. And they're dying of Alzheimer's disease at age 35 to 40. And they look like they're 70, they're getting atherosclerosis and unrelated cancers and diabetes. St. Jude, they follow people for life. So, and we found with them, they did the initial studies, but we found with them that this particular syndrome is associated with accelerated appearance of biomarkers of aging by skin biopsies, for example, accelerated cellular senescence. There are trials underway in COVID. Long haulers looks like it's a senescence-related disorder, for example. There's a trial beginning in chronic HIV survivors who get a lot of these syndromes early, including early Alzheimer's disease. There are trials underway in adult cancer survivors and bone marrow transplant recipients who are getting Alzheimer's disease and other neurocognitive problems, as well as frailty, much earlier than other populations. So bone marrow transplant survivors can get it as quickly as 5 years after the bone marrow transplant because they've had high doses of chemo and radiation. There are trials underway looking at some of the trials are observational. For example, looking at astronauts, it turns out zero gravity will change the shape of cells and can accelerate these processes. And then, of course, space radiation adds to it. One of the reasons a Mars mission has been held off and nobody's gone beyond the Von Allen belt is because, you know, we found with people working in San Antonio and the Houston Space Center and with Brookhaven National Lab that if you simulate cosmic or solar radiation is hydrogen, helium, lithium, you know, 1, 2, and 3 in the periodic tables traveling at the speed of light. Cosmic radiation is things like gold, platinum, palladium flying at the speed of light. It just takes a tiny fraction of the dose of radiation that you would need for smaller particles like electrons, in the case of UV or gamma radiation, to cause cells to become dysfunctional and senescent. There are trials underway in elderly individuals with frailty to see if things will work there. And about 30 of the trials, or 25 or so of the trials of the 87 trials currently in the system are with senolytics. Others are with metformin, rapalogs, sortilin agonists, NAD precursors, and various lifestyle interventions. And some of them are comparing these things. And they're all small trials. They're mainly phase 1. There are a couple of 2As that have been published and one more coming. But the idea is to try to compile the data in some way so that these things can be looked at. And it would be nice if, you know, all the people who are doing trials here, if there's some way of sort of bringing all these metadata together so that things can be looked at in a reasonable way and trying to harmonize some of the data that are collected, that could speed up the way that things work. So in the Translational Geroscience Network, most of the trials are for people with diseases. They tend to have more than one disease. So the kinds of populations who are left out in most disease studies done by drug companies are the very ones that you want to treat. So there are elderly people with 5, 10, or 15 conditions or 20 different drugs. Those are the ones who get excluded from regulatory trials by drug companies. But the population that we deal with are those who have multiple disorders. So the intent is to start off, because many of these interventions, we don't know the safety and tolerability side of things, and there could be considerable risk, is to look at risk-benefit ratios and to take disorders that are arguably related to fundamental aging processes and look at more than just that disorder, but several disorders in the same subjects. And see if they can all be targeted together, or even things like the number of drugs that people are on can be reduced. So the way for particularly the newer agents is to start with conditions for which there is no current good treatment, things like heart failure with preserved ejection fraction or advanced Alzheimer's disease, or other conditions for which there's no current really good treatment because we're worried about safety. And then gradually go back to secondary prevention and maybe one day in the distant future, primary prevention. But you'd have to have zero side effects essentially to really talk about primary prevention in healthy individuals. So a couple of the trials, one of the earlier trials with senolytics of the type that Mitzi mentioned, this is a phase 1 trial with all the problems that these have, learning effects. Everybody in the trial knows who's on the drugs and who isn't, and there are all kinds of biases, but you have to start off with these generally because your main outcome is safety and tolerability as far as the regulatory agencies go. But for what it's worth, in a trial with idiopathic pulmonary fibrosis, which is a senescence-driven disease, and these people don't die of respiratory failure usually, they die of frailty, so they're dying of falls and these kinds of things. That's the clinicians who've seen them will recognize this. They don't usually get to the point of being short of breath, they get frail. So again, given it's a phase 1 trial with all the problems that they have and a very small number of subjects, there appeared to be some signal in looking at 6-minute walk, 4-meter gait speed, chair stands, and short physical performance battery. In the case of senolytics and other drugs, other classes of drugs like xenomorphics are things like metformin or rapamycin or JAK inhibitors like ruxolitinib, things that interfere with the secretory state of these cells. One of the things that the regulators want to see early on is their target engagement. So in the case of senescent cells, you're not looking at a one molecule, one target, one disease approach. This is much more like dealing with an infectious disease. So are you getting rid of the bacterium? And you don't really care what the mechanism is, you care that the bacterium is killed. You could combine multiple drugs like you do for urinary tract infections. Nobody knew how penicillin worked for 30 years, but they were using it. So fully a third of drugs on the market, we don't know what the targets are, the molecular targets. So fortunately, the FDA has bought into this, and it's the infectious disease branch of the FDA that is involved in some of the INDs, and they get it because your target is not necessarily a molecule, it can be a cell type. And again, in subjects with diabetes, obesity, younger subjects, and incipient renal dysfunction, there was some evidence before and after treatment with these drugs on fat biopsies that there was a reduction in senescent cells. That's just an example, but other kinds of agents, we would use other things to look for target engagement. So the first thing is safety tolerability. The second thing is target engagement. After that comes efficacy in small numbers of people, and that's usually 2A trials before you can get anywhere near effectiveness. So some of the trials that are being looked at are a little bit out of what people might normally think. So one of the applications of a lot of these kinds of interventions will be for organ transplantation, most likely. So at the moment, we're throwing away 24,000 kidneys a year because they're from donors over a certain age, even if the kidney looks like it's functioning well. These are, people who sign donation cards. The surgeons will not use kidneys from people over a particular age. With pancreases, they won't use pancreases from anybody over age 45. If we could rehabilitate organs before they're transplanted, while they're on things like life support systems, and if those 24,000 kidneys that are thrown away a year, that would solve the entire kidney shortage in the US. So at the moment, people are waiting 6 to 7 years to get a kidney transplant. They're on dialysis. They usually go through a new AV fistula every 9 months because they clot off, and more than half are dead by the time an organ becomes available. So we're working on this with a bunch of places, and we're hoping to start this with cardiac transplant at Cedars, which is the biggest heart transplant program in the world. We've got trials not actually transplanting the kidneys, but taking human organs while they're on life support systems with the Brigham and Women's Hospital. Stephen Tully is the chief of transplant surgery at Harvard, and liver transplants in Holland, and pancreatic transplants, hopefully, with the Joslin. So when you put kidneys on life support machines, you can, with certain interventions, you can measure what GFR is like on the kidney while it's being perfused. And in the perfusate, you can add things like senolytic drugs, even agents that would be toxic if given in vivo, and clear senescent cells. So in mouse experiments where Stephen Tullius, our collaborator at Harvard, did this, he did heart transplants from old mice to young mice, young mice to young mice, and old mice to young mice, all various permutations. And when you transplant hearts from older animals to younger animals, the animals die fairly quickly, or at least the hearts die fairly quickly, because these are at top. Now he's doing actual transplants, taking out the heart and putting in a new one. You can imagine what the surgery would be like doing a heart transplant on a mouse with an organ that beats at 600 beats a minute. But he started with ectopic hearts in the neck where you can feel them, or use Doppler to see what their function is like. If you treat the donor, the heart itself before it's transplanted, or the recipient with senolytics, we get much better survival. And This can also be done with genetic removal of senescent cells, for example. There are a couple of phase 2A trials that have been published and one more coming out. One of them is not a TGN trial, it's by Unity Technology. And Tamara and I were founders of it early on, but we left it, so we don't have, at least I don't have shares in it anymore, a long time ago. But they did a trial of injecting a fairly toxic, actually, senolytic into the eye in patients with diabetic macular degeneration, and they actually got restoration of vision by 6 lines on an eye chart. We're not just talking about with interventions that target fundamental aging processes, prevention or delay of things. We might actually be talking about alleviation or even treatment of diseases with some reversal of disease-related processes. There are more trials beginning for macular degeneration. There's one with metformin, for example, and there are plans for ones with oral senolytics, so they don't have to be injected. And then there's another one which is beginning with an agent that is a selective mTORC1 inhibitor that is going to be using eye drops. Another phase 2A that was published after the Unity one. The Unity one's only on clinicaltrials.gov. They haven't put a paper out, but their reports on clinicaltrials.gov. The one for age-related osteoporosis was with collaborators at Mayo, and it looked at in that trial. It was found that the people who had the highest senescent cell burden at entry were the ones who responded best, which you'd expect. So biomarkers have to be developed, in our view, that tell you the load, the extent to which the fundamental aging process is operating that you want to target. So we found in the highest tertile, especially, there were the biggest effects. Overall, there was an increase in P1NP, which is involved in bone formation. In the highest tertile of people with senescent cells, at the beginning of the trial, there was an actual increase in radial bone density. So biomarkers of fundamental aging processes, at least our group is interested in, not in just mere aging clocks. Aging clocks that are predictive can be useful in some ways, but I've had, and many of you might have had patients come to you with some biological clock where they're told that they're going to get Alzheimer's disease 3 years earlier than other people. And some of them are almost depressed to the point of committing suicide. It takes a lot of counseling to get them around that. And the only things you can offer are things like, do some cognitive gaming, stop smoking, stop alcohol, those kinds of things, which is advice you give to anybody, anyone. Prepare your family for what's going to come. So there is some counseling you can do, and there might be some value to it, but I worry about the adverse effects of people coming in with these clogs, and they do all the time. Who've been told that they're going to get some disease earlier than other people if there's nothing you can do about it. Where they are useful is, for example, the GRAIL testing, where you can sometimes, high false positive rate, you can sometimes pick up things like stage 1 pancreatic cancer or stage 1 ovarian cancer when surgery is actually curative. But we have to worry about that. In our view, these kinds of assays, the liquid biopsies at any rate, are plus some things like OCT and wearable devices and imaging, especially dental imaging, which looks very valuable. Could be useful in composite scores, provided especially they respond to interventions, and where a response to intervention predicts clinical improvement. So the ideal kind of test that both the EMA and FDA would want in a composite score is something that doesn't just reflect the speeding up passage of the development of these fundamental aging processes, but things that will actually respond in the right direction to interventions. An example of something that doesn't, for example, is GDF15. So GDF15 tracks with biological age, but it goes up even more with certain interventions that arguably might help with health span, like resistance exercise or metformin. Those things make it go up even further. So it might be a counter-regulatory factor, much like cortisol is. If you have someone with septicemia, they'll have a high cortisol. The last thing that you want to do is lower their cortisol when they've got septicemia. That'll kill them. So some of the things that we're looking at may be protective mechanisms that get activated in response to, we have to be thinking very carefully about that. So one of the first biomarker score with respect to senolytics was done by David Allison off that initial target engagement trial. But there are many, many other things beyond this, and there are better things that have come along. But it did correlate very well. It did change very markedly in these very few subjects. In an N of 9, this composite score tracked very well with whether they'd taken senolytics or not, for what that's worth. Same thing with Suzette and Johnny Ward did this with a blood FDG spider test. And then he also looked at composite score biomarkers. Zoledronate is another senolytic, so it's a bisphosphonate which can be injected. The oral bisphosphonates are not very effective because they cause a lot of upper GI symptoms, but zoledronate is given parenterally every 6 months to a year for people with age-related osteoporosis. The group at Oxford, Jim Edwards, Lynn Cox, and others found that, or noted in epidemiologic studies, so this is a good way to try to find these drugs, that not only did zoledronate, which has been used for 20 or 30 years, improve bone mineral density, but it was also associated with decreased mortality, even compared to people who didn't have osteoporosis. Decreased cardiovascular disease, decreased cancers, and improved muscle function after chemotherapy, and increased recovery after infections. So with them, we tested it and we found it's actually senolytic. And it's partly senomorphic as well. It has both effects. Similar thing with SGLT1 inhibitors. They turn out to be partly senolytic and partly senomorphic. So there are ways that you can find these things retrospectively. This just shows that zoledronate is senolytic in some, but not all kinds of senescent cells. So it's not senolytic for senescent human adipose progenitors, but it is for human lung fibroblasts. And it reduces circulating SAS markers as well, using some of these composite scores. So across the Facility for Geroscience Analysis, we're trying to measure the same things across all these trials, and we're storing samples for new biomarkers, as I mentioned, as they appear. It's ideal to collect PBMCs and to do it in the right way, as well as serum and plasma. Serum's important because you can't measure matrix metalloproteinases and other proteinases in plasma easily. And things have to be collected in the right tubes and stored in the right way for these things to work. We found even different manufacturers of tubes. You get different results. So again, I think when it comes to storing these samples, there's going to have to be some uniformity. And the storage conditions have to be right too. Is it liquid nitrogen? Is it -80? Is it -20 in room temperature and so forth? So with some of the blood biomarkers, what we've been trying to do is iteratively narrow down the number of biomarkers by coming up with composite scores, cherry picking post hoc, and then testing in the next trial whether those composites are okay, and then cherry picking from them and using a funneling kind of process to get down to a composite of biomarkers plus things on the physical exam plus results from things like wearable devices or imaging. And then some of these things come up by serendipity. So it looks like one of the major mechanisms by which older organs are promoting senescence in recipients and also decreased cognitive function in recipients is because of production of non-coding nucleotides by senescent cells. So senescent cells are verging on apoptotic, and they can release things like mitochondrial DNA, which get incorporated into cells in an endocrine or paracrine manner, act like transpositional elements, and can induce senescence at a great distance. Even in, if the cells are in the peritoneum, they can induce senescence in the brain. Urine is looking especially valuable. And one of the interesting things in urine with respect to neurocognitive disease is alpha-klotho, which is a geroprotective factor. If you overexpress it in mice, it can increase lifespan for what that's worth. It goes down in Alzheimer's disease. And it's a very large protein, so it's almost impossible to administer it. It's transmembrane protein, but it turns out senolytics make it go up. So we looked at patients with idiopathic pulmonary fibrosis at their urine. We found 20 out of 20 had an increase in urine alpha-klotho after compared to before senolytic treatment. So what we need to do is look for things that are bad that go up and things that are good that go down so that we can develop dimensionless ratios of gerodamaging, or whatever you call them, to geroprotective factors. Then in urine, for example, we're not dependent on creatinine. We could do random urines. And there's a tremendous fluctuation in urine production rate by threefold during the night compared to during the day. So getting dimensionless values will be useful. Cerebrospinal fluid, there are a number of things we look at, and it's beginning to look like, it's very early. The superior nasal swabs will be a good way to get at CSF. So about 50%, apparently, and this is very early, it's Susan Greenfield's work, Baroness Susan Greenfield at Oxford, about 50% of what you get in superior nasal swabs is CSF. And so she's got some pretty good data that are about to be published on that. I won't go through Mitzi's stuff, but she didn't mention during her talk that some of these sphingomyelins and phosphatidylserine and so forth can be measured in the blood as well as in the CSF. That's correct, Mitzi? And some of them, 7 of them, I think you found, sort of may go in the right direction or whatever. So we have to think beyond proteins and peptides and begin thinking of other kinds of reagents. We have to think of new clinical trial designs beyond phase 1, 2, and 3 studies. So adaptive trials, I think, are going to become increasingly important, and that is you measure biomarkers and then you decide on the next agent to give in a personalized way so you know which agent to give, when, and whom, and in what sequence. So a trial was approved by the FDA, which is beginning for people who've had stage 4 or 5 glioblastoma, have had radiation treatment. We know that they're going to get recurrence, 98% of them, and will be dead in 18 months. But what these patients have is an Omiya cistern, so they've got a line with a bubble on the scalp that you can painlessly take tumor blood fluid from. And what radiation and temozolomide, the main treatments for glioblastoma, do is they can make some of the cells become senescent. And so we're alternating senolytics with, in this case, with temozolomide. And now there's a new drug, which was just, a couple of them just came out yesterday in EMBO that we published with collaborators, that looks like it's going to be much better than some of the existing senolytics for glioblastoma. So this kind of adaptive design for serious conditions that are fatal, the FDA will let you do things on the fly, because the way that any physician, if they're doing an operation and they find something weird, they don't have to follow protocol if that kind of condition's never been seen before. So you're allowed clinical judgment. So this particular drug is Buranapant, and there's another related drug, LCL-161, and I won't go through it, but they were mechanism derived, and they're related to things related to P21. I'm trying to finish up. So challenges in future directions. Safety and tolerability come first, and I think clinical trials have to be for that, including for lifestyle interventions. So the military is conducting trials for too much exercise. We always talk about exercise. They're worried about too much exercise, because that can cause microfractures, and the main reason they can't field Marine units, even in active theaters of war, 70% of inability to field a Marine unit is because they've done too much exercise behind the lines. Everything is sort of an inverted U-shaped curve when it comes to food or anything you can think of. We have to think about risk-benefit ratio considerations for new interventions. Some of them we can move fairly quickly, like metformin, because it's been around for a long time, or SGLT2 inhibitors. Others that are new agents, we have to start from scratch. It would be nice to harmonize around the world some of the geroddiagnostic markers and to do this across trials. where feasible. New imaging methods are coming along. I don't have time to go into it, but vibration and viscoelastic things, imaging methods are looking very promising for detecting senescent cells. And that can be done without radiation. New trial methods like adaptive designs and also Laura Getz's N-of-1 designs, where you tell a person not to take something because it's unproven, they insist on taking it. What you tell them is, if you insist on taking it, at least let us get your blood and urine and so forth and measure you before and after. And please stop it every once in a while and restart it. And we'll see if those things change in correlation with the biomarkers and with changes in symptoms. So that's a new math that's been developed. We need to think about combining and sequencing these interventions. Very importantly, we'll be combining gerotherapeutic and disease-specific interventions. This will be very important in my view for heart failure with preserved ejection fraction, scleroderma, lung fibrosis, liver fibrosis, kidney fibrosis, where it's looking like TGF-beta active drugs like losartan in combination with senolytics. Either drug alone has maybe a 20% effect in preclinical models. The combination has an 80% effect, so it's much more than additive. But we don't know that in humans yet. And the real question is, how do we proceed fast? Because people are dying with some of these horrible diseases. Like I hate glioblastoma, I hate triple negative breast cancer, especially the BRCA negative form. But at the same time, responsibly, because anything that sounds too good to be true is. There are serious side effects with a lot of these things. How do we manage expectations of the public? How do we stop some of the hype that's going on? And how do we do this in an academically responsible way that's evidence-based, but do it quickly? And so those are real challenges. So I'll just conclude by thanking a lot of people, including Mitzi's in the audience and many other things.
Does anyone have a question? Well, oh, you don't have to apologize. We love hearing from you. Any questions? Right.
I had a question about the biomarker development in these intervention studies. So I can understand, if you have senolytics, you can develop biomarkers for that particular pathway. But if you have agents that are targeting other pathways, you anticipate that you would have biomarkers that can span?
Yeah, we're in the TGen where we're not only 25 or so of the trials are with senolytics. So we're measuring things like NAD related molecules up and downstream things. Things are downstream of mTOR and looking at TOR activity roughly, by looking at phosphorylated TOR and looking at TORC1 versus TORC2. Looking at some of the things that get produced that will affect NAD, for example, CD38 on macrophages, which degrades NAD, looking at sirtuins, so we're looking at clotho, which nobody knew was anything to do with any of these things. And hopefully we're going to be adding various steroids. So high doses of glucocorticoids, for example, induce senescence, alpha-estradiol is protective, so we haven't added those yet. We need to. And then things like Mitzi found, for example, that may be related to myelination that get into the blood, like some of the sphingomyelins and so forth. So there are going to be disease-specific things that are in these age-related diseases and disorders. You obviously want to know what glucose tolerance is like and that kind of thing. But also, particularly for as many of the hallmarks that we can measure as we can. Because if we want to test, is the unitary theory true? If you target one, do you affect the rest? And also, we would only want to give drugs, and the FDA insists on it for certain trials, like the St. Jude trial with childhood cancer survivors. They'll only let us give the drugs if we measure senescent cell burden in the blood. We have to show it's above a certain threshold before we're allowed to enroll.
All right, thank you so much. Up. Okay, the next speaker is Aleksandr Tyshkovskii. He's going to be talking about transcriptomics.
Thank you. Good afternoon. It's a great pleasure to present our work in front of you. I'm an instructor in Harvard Medical School representing Gladyshev Lab on this meeting. And I want to start from a global idea. So when we talk about molecular clocks in general, of course, first of all, they should work and should be validated. But after that, there are still some criteria, some features that are desirable when we talk about molecular clocks. And now we see more and more universal clock coming in the field. And of course, one of the best examples of such clocks is a pan-mammalian clock developed by Steve Horvath that was trained across different tissues, across more than 300 mammalian species. And it's very applicable because of that. Even if you have new tissue, you can still apply it. So it's very useful in new setting. Another criteria is a criteria of interpretability. And I think the very nice example of interpretable clocks is proteomic clocks, because we know, at least at some level, we know function of individual proteins. So we can look at top proteins that contribute to the effect of intervention that we observe. And also using molecular data, we can develop not only composite clocks, but also clocks trained on subsystems. And in terms of proteomic clocks, this would be, for example, different organs. And this idea was first used by Tony Viscari. And in our lab, we were inspired by this idea. And Tony Viscari developed chronological clock that were organ-specific. We used the same idea, and using UK Biobank, we developed organ-specific mortality clock that predict time to death using proteins that are produced by specific individual tissues, for example, by brain or by liver or by lung. And as you can see on this plot, all of them predict overall mortality because we were trained to predict overall mortality. But when we look in more detail at incidences of individual diseases, we see that, for example, liver clock predicts liver cirrhosis and fibrosis better than mortality in general, and lung clock predicts COPD. So such approach can give us more idea about the mechanisms of particular interventions, which particular organ, which particular subsystem in the organism they affect. And here are just a couple of examples. Here you can see the effect of smoking and the effect of drinking. And in case of smoking, it's very consistent across different organ clocks, but the highest effect is observed for lung, which again is consistent with our expectations. For drinking, it's even more interesting. We can see that some clocks show significant proaging effect, like intestine and brain clocks, but our clocks, on the opposite, on the contrary, they show even anti-aging effect, for example, artery clock. And this is consistent with some studies showing that low doses of alcohol may actually slightly reduce the risk of cardiovascular diseases. So such subsystemic clocks can give us more ideas about heterogeneous effects of interventions on aging. So we see here that aging is not homogeneous. It includes different systems, and the effect on the same intervention may be different when we look at different subsystems. And the final feature I want to mention is a feature of applicability. Of course, if we have lots of molecular data for certain omics, if clocks can be applied to single cell data, if they're cheap, it's also a nice feature. And when we looked at this criteria, we thought that transcriptomic clocks could actually fit them very nice, because for RNA-seq, it's one of the most routine procedures. We have lots of data for many species, many tissues, and they are also interpretable because we know functions of individual genes. So we decided to go deeper into this topic, and we started from collecting data for mice and rats. There are lots of publicly available data sets, and we aggregated data from 96 data sets that cover 26 tissues of rodents, different ages, and using this data, we trained chronological clock. So this is very general composite clock. You can see that the quality on test data set is pretty high. It's more than 0.95. So it's comparable with epigenetic clocks. But besides, we also collected data from animals subjected to different interventions that either extend lifespan or reduce lifespan. So for example, we have progeria models, or we have certain lifespan extending drugs like rapamycin, for example. And for each of these interventions, we also collected publicly available lifespan data. For example, on the right, you see a lifespan study from ITP that shows lifespan of mice, control mice, and mice treated with rapamycin and acarbose. And when we have lifespan data for every model, knowing its age, we can predict expected mortality, right? We can feed survival data with common models, like for example, Gampert's model. And then for every sample, we can not only predict its age, but we can also predict expected mortality of this particular animal based on its interventions, based on its strain, based on its sex, etc. And so we did that, and we trained mortality clock that predicts expected mortality not only across normal mice of different ages, but also across mice subjected to different interventions. And you can see that quality is also pretty nice, and we validated this clock by checking how it predicts the effect of interventions with no effect on lifespan. And indeed, our clock predicted lifespan shortening interventions to have higher effect on transcriptomic age, and lifespan extended interventions, they had reduced transcriptomic age. So this is composite clock, but of course, the more general question would be if these clocks can be applied across species and across cell types. And we decided to start from single cell data. We utilized Tabula Muris dataset that includes gene expression data on different cell types from mice of different ages. And we used meta-cell procedure, aggregating individual cells into so-called meta-cells to increase coverage. And when we looked at individual cell types, we saw that actually clocks, although they were trained on tissues, not on cells, but they still can capture aging of different individual cell types. So the same model was applied to different cell types, and we can see that for them, it predicts significant age-related change. And then we expanded this analysis to all cell types that are available in Tabula Muris dataset. We observed that for almost all cell types, we see significant increase of transcriptomic age with time. We also looked at individual genes that are most contributing to pro-mortality effect we observe. And many of them are related to inflammation regulation, to apoptosis, but also there are several interesting genes. I highlighted two of them. One of them is galectin-3. It's a pretty interesting gene because it regulates not only inflammation, but also proliferation and even insulin signaling. So it has many different functions. And also CDKN1A, it's P21, famous biomarker that's used for tracking senescence, for example. And you can see that they increase their expression with age across different cell types, so they're very consistent biomarkers of aging across individual cell types. We also decided to expand the analysis across species. So the clocks were trained on mice and rats, but we also aggregated data on humans and on monkeys. And first, we just compared general gene expression changes with age and observed that they are positively correlated across species. So it's a good sign. It means that we can now train multi-species clock. And that's what we did. And here you can see the quality on test set for 4 species. So it's the same model applied to different species, 4 species. And you can see that in each case, it captures aging dynamics and captures expected mortality. So now we have transcriptomic clock that can be applied across species and across tissues at the same time. Now I want to go into interpretability side. So here I presented composite clocks, but similar to proteomic clocks, we can go into more detailed level. We can look at subsystems. And to do that, we performed network analysis and we identified modules of genes that are correlated. So they change their expression at the same time. We identified about 26 such modules that were correlated during aging and in response to interventions. And interestingly, when we looked at functional enrichment, we saw that they are enriched for particular very distinguished functions. So for example, we have a cluster of genes enriched for inflammation and cluster of genes enriched for metabolism, etc. And now using each subset, we trained module-specific clocks of age and mortality. So these clocks predict age and mortality based on genes associated with only one particular pathway, for example, inflammation or metabolism. So now we can track aging of individual subsystems within the cell. And on the right, you can see that mortality clocks predict both age and lifespan, so they can be applied in both cases. And of course, the accuracy of such clocks is lower than the accuracy of composite clocks. But the advantage is that we can use them now to, again, identify subsystems that respond the most to particular intervention. And I want to just show you some examples how it works. So first, we looked at the effect of caloric restriction. And here you can see that many module clocks show anti-aging effect in response to caloric restriction, we expected. But also you can see that top modules, top subsystems, are modules associated with metabolism. Heme metabolism related to mitochondrial function, lipid metabolism, cholesterol metabolism, etc. And we know that caloric restriction affects metabolism, of course. On the other hand, if, for example, mice are subjected to lipopolysaccharide injection in their brains, we can see that in general, there is pro-aging signal, and mostly it's driven by inflammation module. So again, they have validation that modules predict what they should predict. Then we were interested to apply these clocks to human data, and we got access to Framingham Heart Study and checked if module clocks predict mortality, time to death in people, based on blood data. And they saw that in general, they have positive association with mortality, but some modules, as you can see, have high association than the others. And you can see that in general, transcriptomic clocks are on par with epigenetic clocks. Epigenetic clocks are shown in blue and transcriptomic clocks are shown in green. But among transcriptomic clocks, clocks associated with inflammation, chromatin modification, and oxidative phosphorylation show the highest association with mortality. And then we looked at correlation between transcriptomic clocks and epigenetic clocks after adjustment for age and sex. We observed that, first of all, on average, they are positively correlated, but among all transcriptomic clocks, chromatin modification clocks had the best correlation with epigenetic clocks. And again, it's very nice and consistent with our expectations. So here, in rows, we have transcriptomic clocks. In columns, we have epigenetic clocks. And you can see that on average, there is positive correlation, but the most correlated transcriptomic clock is chromatin modification. And interestingly, we can see that it's correlated mostly with second generation epigenetic clocks, such as FENA-H and dunedin pace, which suggests that these clocks capture indeed some biological features, not just chronological age. So now I want to double shift gears and talk about established models of chronic diseases and models of rejuvenation. Of course, to apply clocks, we should validate that the clocks predict status of diseases, problems with health. So we used several models of chronic diseases in rodents. We also have data in humans, but I don't have time to present it today. I will present rodent data. And here you can see that the data sets are relatively simple. We have mice, control mice, and mice with certain disease models of the same age. And we compare the transcriptomic age between control and diseased mice. And we can see that different diseases, we have Alzheimer's disease here, chronic kidney disease, ischemic stroke, etc. Most of them have significant increase of transcriptomic age. So they elevate transcriptomic age, which is expected. And when we looked at individual genes contributing to this pro-aging effect, we saw some very conserved biomarkers. So for example, we saw that CDKN1A and galectin-3, which I already showed you previously for individual cell types, they are positively associated with mortality across different diseases, different chronic diseases. But also what is very interesting here, you can see that one disease, which is hepatocellular carcinoma in right lower angle, right lower corner, it has negative effect on transcriptomic age. And we were puzzled with that. We have cancer model, the cancer tissue itself, but it shows lower age. Why is it so? We looked at modules that are responsible for that, and we saw that two modules the most contributing to its anti-aging phenotype. And these modules were models associated with cell cycle and differentiation, EMT, epithelial to mesenchymal transition. On the other hand, you can see that other modules, such as inflammation modules, they show pro-aging signal. So again, we have very clear example, then the same model, the same intervention can have both pro-aging and anti-aging phenotypes. And only looking at subsystems, we can distinguish between these two features, these two trajectories. So then we looked at the models of regeneration. And one of the most common model of rejuvenation is a model of induced pluripotent stem cells. So this is human data, and consistent with epigenetic clocks, we see rejuvenation induced by reprogramming. And most module clocks also show rejuvenation, but The top clock is a clock involved into differentiation, which is again consistent with the literature. We also applied our clock to the model of heterochronic parabiosis. This is a very interesting model where old mouse is attached to the young one. So circulatory system of old and young mice are combined. And actually, there is lots of literature showing that old mice get some health benefits after this procedure if they survive. Actually, the survival rate is very low. But if they do, then they are very healthy. They have high level of stem cells, lower inflammation, etc. And in our experiment, we connected both young and old mice either to mice of the same age, isochronic parabiosis, or to the mice of different age. And here you can see that if young mice are connected to old ones, their age is higher, transcriptomic age. But after detachment, after 3 months of detachment, actually the transcriptomic age doesn't differ. So they recover, young mice recover after detachment, even if they were connected to the old mouse. On the other hand, old mice connected to young ones, they are indeed rejuvenated. And what is very surprising here, even after detachment, this effect still maintains. And also we saw actually in the same experiment, we saw that old mice connected to the young ones, they had higher lifespan, so lower mortality. So it's consistent with the data we see here. And also we saw that many module clocks were associated with this effect. Finally, one of the most counterintuitive models is a model of early embryogenesis. So it was first hypothesized by Dr. Vadim Gladyshev and then supported by epigenetic data that during early embryogenesis, there is a period of rejuvenation, and only then we see very monotonous pro-aging signal in animals. And this is data for mice, epigenetic data. And we decided to check the same at the level of gene expression. And indeed, when we looked at transcriptomic age of mice during embryogenesis, we saw consistent with epigenetic data that first we see rejuvenation, and only approximately after day 10 of embryogenesis, we see monotonous aging. And actually, there were several modules associated with this effect. They included modules involved into inflammatory signal and modules associated with metabolism. So these were two main groups, and you can see that they show U shape. First, there is rejuvenation, and then there is aging. But there were other subsystems that didn't show such trajectory. For example, here on the top, you can see inflammation module. It shows very consistent behavior, U shape. But below, you can see cell cycle module. And you can see that it shows monotonous aging right from the beginning of embryogenesis. And there is no rejuvenation in any period of embryogenesis. So again, we decided to check if there are any consistent biomarkers of rejuvenation models, and we found them. In general, mortality-associated changes induced by models of rejuvenation were positively correlated with each other, but top biomarkers were, again, galectin-3, CDKN1A, and also S100A9, which is also a biomarker of inflammation. So we see that same biomarkers again and again appear in different settings, different models, in cell cultures, in tissues, in mice, and also in humans. So we decided to check the association of these genes with mortality in human data using FHS, Framingham Heart Study. And we saw that many of them, especially galectin-3, showed very strong positive association with mortality and incidence of cardiovascular diseases. And the same was supported by UK Biobank data, where we looked at concentration of these proteins in plasma. And again, galectin-3, among with other genes, showed strong positive association, not only with mortality, but with many different diseases. You can see the list on the right, and it includes heart failure, kidney failure, type 2 diabetes, and even some established biomarkers, like, for example, obesity and hypertension. So we can see that indeed, galectin-3 is one of the most universal biomarkers of aging, mortality, diseases, and rejuvenation, both at the level of gene expression and at the level of proteomics, which is very interesting. And of course, we don't know if it causes mortality or if it's an adaptive response, so we will check it now in experiments on mice, but this is a very interesting candidate. So with that, I would like to summarize my talk. So we discussed features that are very desired for molecular clocks, and transcriptomic clocks seem to meet most of them. So we can develop universal clocks that work across tissues, across species. We can use them to identify the effect of novel interventions that either rejuvenate or modulate lifespan of animals. And also, these clocks can be applied to single-cell data. We can use module clocks to get better idea about the mechanisms of interventions, to look at specific subsystems of the cell. And finally, we can use them to identify the most conserved and most contributing genes, specific genes that explain the behavior we observe. So we believe that this will help the field, and transcriptomic clocks will be broadly used by different groups. And of course, will be happy to collaborate with all of you. And with that, I would like to mention that most of the data I showed you is already available in the preprint. Not everything, but most of the data. And I would like to thank our lab, Dr. Vadim Gladyshev, our amazing collaborators, and of course you for your attention. Thank you.
For a question?
Great work. I'm glad there's a paper because I need to read it.
Yeah.
I think my understanding with transcriptomics was it was quite difficult to apply in big studies. And that's why the clocks haven't really taken off till now. But is your clock applicable in different transcriptome data sets?
Yeah, that's a great question. So one of the biggest problems with transcriptomic data is that it's very subject to batch effect, and it's typically very hard to translate from one dataset to another. It depends on platform, even on the laboratory, and everything. So to kind of get rid of this problem, we applied some advanced normalization techniques that I explained in the paper. We can also discuss them later. But also the other important feature we used to train the clock, we used many different datasets. So we used more than 100 datasets. And this kind of ensured us that the clocks is broadly applicable. If you train the clock, transcriptomic clock, using only one dataset, most probably it will not be translated to others. So that's the problem we observed. Yeah, but now, well, it seems to work in general, so. Yeah. Also, I should mention that in animals, actually, it works much better than in humans because human data is more noisy because of because of behavior. For animals, you can control their behavior. For humans, it's impossible. It's also a problem.
Thank you so much.
Thank you so much.
Okay, our next speaker, our last speaker before the break is Brendan Miller talking about, I'm assuming mitochondria. I don't even have to look at it.
Okay. Is too loud? Yes, it is.
No, it's good.
Well, first off, thanks for inviting me. I did my graduate schooling here, so it's fun to be back. We've been new building across the street. Things are changing. It's hotter in here. The air conditioning needs to be turned up. Just saying. But we'll get through it. So today I'm going to tell a story more so about just how genes and protein coding genes are characterized and annotated. So I want to keep this in chronological order. So we'll start with the year 2000 and the first few slides. We'll finish with the current state today. A lot of the work will be stuff I did during graduate school. Some of it will be what I'm doing now as a postdoc. But in general, it's all about trying to find these small genes, which if there's any one takeaway, a lot of it I'll say is probably going to go, beyond you won't remember it. There's one thing to take away is that protein coding gene annotation is really good, but there's still significant room for improvement. And so the goal of this talk is to kind of make that clear, as well as the methods we're using to make that better in an attempt to give a lot of omics researchers like yourself more gene elements to see if they're important. That gives us molecular biologists, biochemists more incentive to study the function of these genes. And because a lot of these genes make small peptides, well, they pose as potential drug candidates as well. So some of the jargon will be new. I don't want to get too jargon heavy. So I'll try to keep this pretty general, but that is the takeaway line that I get. So if you start with the year 2015, GenCode, which is a common gene annotation database we all use had about 200,000 annotated transcripts. And then if you fast forward 10 years later to today, that's almost doubled, which is pretty significant if you think about that. The expansion of transcript is nearly doubled in just 10 years. So the question is, why did this happen? That's because of more long read native transcript sequencing, more tissues, more investment, and it's quite astonishing to see such a big increase. Now, if you compare the other commonly used reference database, which is UniPro, any proteomics person is going to go to UniPro, go to the reference proteome tab down with the Homo sapiens reference proteome, and at the time in 2015, there was about 90,000 proteins. You fast forward to today, and it's about 105,000 proteins. So it's about a 17% increase. So then the question is, this separate increase of transcripts versus proteins, is that biologically meaningful, or is it just that protein translation is diverse and controlled at the transcriptional level? And that's a question that my research attempts to address, as well as other labs currently talking about the same topic. But we were cautioned against this in 2004 when the Human Genome Project actually published their landmark paper. And hidden in the discussion was an acknowledgement that while we have about 20 to 25,000 protein coding genes, there's a segment of protein coding genes that might be missed. And these tend to be small, potentially fast evolving, some of which are still conserved but fast evolving, but they tend to encode these small proteins under 100 amino acids. And this has persisted up into today. So I'll come back to this slide towards the end, but this is important because a lot of this technology we use is geared towards finding larger proteins and not these small proteins. And so if you fast forward again, getting the Millennium Falcon to today, you can see this etched out just in UniPro. So again, go in, you download every single sequence from UniPro, and you see this cliff. And this cliff starts at around 0 to 100 amino acids, of course, to start. And the majority of proteins annotated are between 100 amino acids in length all the way down to 800. So of this red sphere, are we missing some, or is this again just biologically, natural, right? And that's really the core focus of my research, as well as many of us at the Salk Institute. So really for us, we want to fill this red gap. So we think this red gap is important. We have a lot of data and history to suggest that we are missing these small proteins. We talked about a lot of neuroendocrine biology today. A lot of texts we use were actually founded by, which I'll get into it, Wiley Vale and Roger Gielman, of whom characterized the canonical neuropeptides such as myostatin, TRH, CRF, using a lot of the same biochemical extraction techniques that we now apply to finding these new, what we call microproteins. Full disclosure, the word microprotein is kind of a stupid name. I don't really like it. It's really just any protein under 150 amino acids. The challenge we have in the field is that illustrating this specific concept is difficult to express. So if you went to the Keystone Conference for microprotein 2 years ago, that was a hot topic. What do you call these things? They can have many different names. It can be small peptides, mini proteins, microproteins, etc. The only thing you need to understand is that these are proteins under 150 amino acids. That tends to be the default definition today. And so while we want to annotate and find these microproteins, we also want to count them, make good databases to deliver to a lot of folks generating good algorithms, clocks, implementing transcriptomics-based methods. But we need to do a better job of finding these genes. And ultimately, one of the goals for me is you find the genes, you disperse them to you guys do great work. We characterize them, the biology, and maybe some of them are posed as good drug candidates, and you can engineer them with some disease relevance. And so what is a microprotein? What are the genomic elements that encode these? I don't want to get too much into the detail here, but this cartoon is representing the diversity of where these small open reading frames exist spread throughout your transcriptome. And so colored in dark gray is what you would see from UniPro, those dark gray coding sequences. Now, microproteins are coded by these SMORs, or the gene elements that make these microproteins in blue. And you can see they can be spread all throughout one transcript. One can be upstream of a canonical coding sequence, a UORT, for example. One can overlap a coding sequence. One can be downstream of a coding sequence. And then you get all these other different jargon type names. We won't go into it, but if you're curious, we call these ribosome intronic ORFs, non-canonical ORFs. These are transcripts by GenCode that don't have no protein coding annotation, long non-coding RNA ORFs, which are kind of misnomers because some of them are coding. And then we have these pseudo-ORFs, which result from a reverse transcription event, basically a gene duplication event. They're actually quite interesting and a hot topic for our lab community. Okay, so in 2015, the field kind of started to take launch, I guess. You can call it a big bang, if you will. And if you look at just the PubMed citations for the keyword microprotein, it flatlined from 2000 to 2015. And really one of the first microproteins discovered actually came from here at USC. This is a picture, I think, of UHASI from the year 2000, I believe. Got hair number one.
Graduation.
There you go, there you go. And one of the first microproteins, or really small proteins, we call it mitochondrial peptides, was humanin. This thing is one of the smallest Swiss reviewed proteins to date. And then soon thereafter, the human genome was sequenced. It was published in that 2004 Nature paper that we just talked about. And then in the late 2000s, there's a surprising discovery that if we go a step further in transcriptomic sequencing and we just pull down ribosomes, so you enrich for ribosomes, and you sequence the embedded RNA within a translated ribosome, a lot of the reads mapped to non-protein coding regions. Now that could be just dysregulation of the protein coding gene, or it could be non-canonical or potentially unannotated new protein coding elements. So that was quite surprising. Soon thereafter, a few years later, my current lab, Alan Secretarian, developed a proteogenomic technique where you take a transcriptome, you annotate every theoretical small protein frame, every potential microprotein, and you search for it in mass spectrometry-based methods. And what was found were several hundred microproteins, but this, which I'll get into, was a little bit more surface up, but it showed that by taking a transcriptome, just re-annotating the protein coding potential, you can find several 1 to 200 unannotated, uncharacterized microproteins that were not, of course, annotated. Then this big bang happened, and in 2015, citations went through the roof. They took these two methods, ribosome profiling, which are sequencing RNA, and then the proteogenomics, and other labs started to adopt this. We adopted a segment of this in Haasi's lab when I was a graduate student, the proteogenomics side, and we started to see a lot of these transcripts that were previously annotated as non-coding in databases actually encoded a protein. And so one example is MOTC, which was published in 2015. We found a brain-specific one a few years ago called SHMOOZ, and this you can see the rapid aging of Haasi right here. And then in 2023,
25 years.
That's hardly rapid.
So, in 2023, the field started to recognize, okay, we have all these different techniques. And so let's bring together a consortium where we combine a lot of our ribosome profiling or proteogenomics, and let's try to annotate these microproteins once and for all. And so that's where we currently are at. I'll take a quick segue into just how we implemented more of a proteogenomics approach to find SHMOOZ, then we'll get into more of the nuclear encoded microproteins. But SHMOOZ went from discovery in 2017 in terms of detection using mass spectrometry to Swiss Pro reviewed in 5 years. It's actually quite a fast timeline. The reason this is actually pretty fast is because we implemented in 2018 more of a population omics perspective. So exactly what we did is we're able to re-annotate the mitochondrial transcriptome and search for potential peptide byproducts. One of them came up was what we eventually call SHMOOZ, but then we mapped genetic variants in the mitochondrial genome back to these peptides. And one of the published at the time in 2011 SNPs that predicted in ADNI, Alzheimer's disease, overlapped the SHMOOZ gene. So that gave us more reason to clone this, synthesize it, and study its function. We went on with Eileen, with Art Toga, with David Bennett at Rosmap. to fully examine the phenotypes of the same SNP and validate that SNP, and also went into more transcriptomics databases and try to understand the correlation of this transcript or schmooze expression with other genes for functional experiments. And we were able to functionalize this as an intermitochondrial membrane protein as maybe one of potentially other functions. That led to us getting this Swiss Pro reviewed, which we don't do anything. This gets annotated up and deposited in the database in 5 years. So we would like to do this for, ambitiously every microprotein. And so this is an example. This is probably going to go right over most folks' head, but in the middle is for this case, mitochondrial genome, every potential peptide. And on the outer ring is 13 mitochondrial encoded proteins. You have, it depends how you annotate it, maybe 400 to 800 mitochondrial derived peptides or mitochondrial encoded microproteins. Now, specifically for schmooze, just very quickly, again, we were able to go back in and analyze this SNP that overlapped the schmooze gene and increase Alzheimer's disease risk by about 30%. With Arthur Tobin's group, we were able to go in and look at this effect in UK Biobank in 40,000 Europeans. And the same SNP correlated with hippocampal atrophy over time. And then finally, we were able to get high quality spectra with now my current lab at Salk Institute and Aleksandr Tyshkovskii's lab by enriching for this microprotein and using specific biochemical techniques to essentially get rid of larger proteins to enrich the sample with small proteins like schmooze. Now, schmooze, generally speaking, if you give it to cells, it's going to induce a mitochondrial transcriptome response. If you give it to rodents, for example, ICD in rats, it also induces a mitochondrial transcriptome response. And if you look in human beings, post-mortem brains from the Mayo Clinic, that same SNP that associates with hippocampal atrophy as well as Alzheimer's disease, it actually deviates those individuals away from the norm, and those genes that permit that deviation map back to mitochondrial transcription as well as protein synthesis. So you have this harmony across experimental as well as an omics, population omics perspective. And again, this is the Swiss Pro review and kind of a template, if you will, of what we hope to do. So this type of work, these microproteins, as I mentioned earlier, is gaining focus, and it requires a lot of different data sharing. And what I mean by that is to do a ribosome profiling experiment or do proteogenomics takes time, it's really expensive, and you're limited by a cell type or a tissue type. That can take years to do. So what this group in collaboration with Genco did is combine from across several different labs, of course, one of which is us at Salk, all of our existing ribosome profiling and proteogenomics data to create this high quality SMORE for microprotein data set that then can be shared to omics researchers, and you can count those genes in a lot of your analyses. And what was found was that there's 7,000 actively translating SMORFs. This means that this SMORF that may encode a microprotein, or may just be a cis-regulatory element of a protein coding gene, there's 7,000 of these are being actively translated. Now, Swiss Pro currently has about 20,000 annotated proteins. So we have 7,000 actively translated SMORFs. That represents almost 40% increase in the potential amount of annotated proteins. Now, the reality is, or the hypothesis is that not all 7,000 of these are making proteins, but perhaps maybe a fraction of them are, and it's up to the community to determine which those are. Now led to a machine learning approach that we developed in our lab that we call SureStep, which essentially takes all of that data, the ribosome profiling data, and it asks a simple question: which of those translating SMORFs look like a Swiss Pro small protein? It's a very basic question, and we use a way to do this is to generate this negative training set. Now, the problem is we don't have a robust negative training set for machine learning development. So, for instance, I mentioned a long non-coding RNA. It's a misnomer. Over the past 10 years, there's been dozens of long non-coding RNAs that actually do encode a protein. So if I include that in my training data set as non-coding, well, I'm going to get not accurate predictions. So we need a robust way to actually have this gold standard negative training set, which currently doesn't exist. but we need to start somewhere. So what we did was we took all the translating SMORFs and we took the amino acid composition, we took the length distribution, we took the nucleotide composition, and we in silico translated a negative library randomly. So we have a randomly generated now set of microproteins to which we can compare those that have been experimentally characterized. And we were able to test many different algorithms, yada, yada, yada. The point being is that if you look in the top right, you can kind of see this in reduced dimensional space. So if you look at a UMAP, the navy dots represent that randomly generated microprotein set. Now, if you overlap the features of those actually characterized as Swiss proteins, in yellow and teal, the yellow represents intracellular small proteins or microproteins, the teal represents secreted ones. And you can appreciate that there's separation between the secreted and these intracellular, but also from the randomly generated ones. Now, if you overlap in gray, these the mudge or the translating SMORFs, most of them overlap with the randomly generated SMORFs. This means that a translating SMORF looks like a randomly made protein I made in my computer. But there's several hundred that actually do share biochemical feature space with those that are characterized. And we did this in collaboration with Hosang Kim at USC. It's quite accurate, has about 93% precision, and 88% recall in testing this on holdout data from Switzerland. So here's an example of how we can apply this. So if you do proteogenomics and you take, in some cases, I'll show you an example in the brain, but some tissues have 5 million potential peptides. If I throw that into a database for a mass spec search, I'm bloating the database. I'm going to get back a lot of false positives. So I need to find a way to take this data back and pick which to study functionally. So this is where I can throw in this algorithm, where I can find, okay, I have a hint of mass spec detection. Now, which ones look like experimentally curated proteins? And one is encoded by this upstream open reframe by a gene body called STAR B1, or it's a mitochondrial transfer protein, and we were able to generate antiserum against this and show that it's actually quite abundant in reproductive tissues as well as in adrenal, in which you see the STAR transcript expressed. But we're able to do this by biochemically enriching these samples, which we'll get into in the next few slides. So if you look at now specifically the brain, which will be the last, what do we got here, about 8 minutes of this presentation, we wanted to apply this to finding new brain peptides. Again, basic goal. And so what we first did is we used a long read transcriptome or nanopore-based transcriptome, and we just tried to find every theoretical SMORF, any theoretical SMORF that might make a microprotein. After we did that, and by the way, half of which of these samples were Alzheimer's disease, the other half were just convally intact age-matched controls. Then we were able to search for those using deep TMT-based proteomics in collaboration with Rush Alzheimer's Disease Research Center and the ROSMAP study, and use those reads and map it onto potential function through our short-stop algorithm. And what you can appreciate is that first on the bar graph, on the x-axis is a number of SMORFs or potential protein coding genes. Swiss probe is only about 1,000, again, small proteins, right? So that big red gap, that's represented right here in this bar. Now, if you just look and ask the question, well, how many of these SMORFs are in this brain transcriptome? It's in the millions. And in fact, most of these don't look like a Swiss probe protein. If you throw over the short-stop algorithm, a sliver do look like a Swiss probe protein that's characterized, which is interesting. And a lot of these don't even have a canonical start codon. So not even ATG start codon. That is interesting for maybe cryptic protein expression that you might see, like I say, ALS, even Alzheimer's disease. There's a lot of these SMORFs. Again, how do we figure out which ones of these are making peptides? And so when we now overlap that and search for them using TMT-based proteomics in 600 brains, we're able to essentially detect most of the brain-expressed microproteins by Swiss probe. That's good. Now we're also able to detect almost 3 times more small proteins or microproteins using our search database. And in color here is a resemblance to the Swiss pro, and this is the shortstop classification. We actually see almost a doubling of novel proteins, microproteins, that resemble in reduced feature space that Swiss pro protein. We also see some dissimilar ones in navy blue and some non-ATG ones in gray. Again, the question is, well, what are these things doing, and are they even important? And so we can quantify these, and in red, again, are all the experimentally characterized ones that you can download, and in gray, yellow, and gold are the ones that are novel. And like how we quantified a lot the Swiss pro ones, we can quantify a lot of the novel ones, and they're actually differentially expressed. On the top right is MDK mitokine. This is a replicated result from the 2022 Johnson paper using this data. It's crazy how far away on the top right that is. But again, the question is, well, what are these things doing, and can they actually be looked at the RNA level as well? So we went in and we matched the RNA-seq to all those 600 brains from ROSMap. And if you look at MDK, you only see a difference at the protein level. So at the RNA level, you're not going to see this MDK change. You're only going to see it at the protein level. If you look at now our novel proteins, the same thing happens. We have a novel microprotein expressed by this RAB3C gene body. You only see it at the protein level. And then if you look at these pseudo-ORFs, you will never or rarely count these pseudo-ORFs. These are gene duplication events. If you take a nucleotide sequencing workflow, they're multi-mappers. They get discarded. So at the RNA level, because they map multiple regions, you will not be able to find this in transcriptomics. You only find this in uniquely tryptic fragments from mass spec. And again, we were able to detect this using mass spec, and we see marginal increases in AD brains. Now, here's one example of the importance of having this information for omics-based studies. So what tends to happen is we use the GenCode GTF, we count all the different coding sequences, and we collapse it into the gene level. But there are instances like in this MKKS gene body, where if you just collapse the gene body into one value, you're going to miss this at the RNA level. And if you look here on the bottom right, in my mouse goes, there we go, there's a smaller coding sequence that at the RNA level is lower in Alzheimer's disease brains. Now, you go to Swiss pro, this is not going to be in Swiss pro. You go to Swiss pro, this is in Swiss pro. So if you count this, you're going to see no difference. If you count this, you start seeing a difference. At the mass spec level, interestingly enough, you don't even detect this at the mass spec level. So this is annotated as the canonical ORF, but you have no evidence of detection at the mass spec level. The only evidence of detection is this smaller coding sequence upstream that is also lower in Alzheimer's disease brains. Now, when you clone this, overexpress it, it localizes to mitochondria. A lot of these small proteins tend to be mitochondrial based. And now this is one program in the lab where we went from a in silico computational approach down to a disease relevance approach down to functional studies going forward. And so what I'm going to leave here in the next few minutes is how you can also apply these characterization algorithms to make functional hypotheses. So one question is, well, what about cryptid proteins? So if you look at like a lot of aggregate-prone proteins, like an ALS, C9orf72, it has a near cognate start codon. So it doesn't have an ATG start codon. You've got these repeat elements, it forms this ribosome entry point, and you get these aggregate proteins. They tend to aggregate up. As we know, a lot of these dementias, they tend to be aggregate proteins. It's a protein family. And so if you look at now extracting laser dissecting out plaques from postmortem Alzheimer's disease brains, and you take the nearby region of these plaques and compare the enrichment pattern of the plaque for proteomics versus the nearby region, you're only seeing about a few Swiss pro small proteins. Now, if you overlap the classification of the entire microproteome, we see basically enrichment of small proteins that don't look like Swiss pro proteins and non-ATG proteins. And we actually see it almost flip, no plaque specificity versus plaque enriched or depleted. Now, frankly, I don't know what the depletion means. It is nearby the plaques, but this is hinting that the plaque that we look at, which is amyloid beta, reactive microglia, other proteins, it could also be a byproduct of this cryptic type expression and these gene bodies that are worth studying. So although we want to find microproteins or small proteins that look like functionally relevant ones, it's also necessary to look at the ones that may slip into translation, be aggregate prone, and maybe contribute to that proteinopathy that you see in dementia. The problem though is that you can't quantify these things. So if you look at, again, 600 brains, you ask the question, well, how many of these Swiss proteins can you even quantify? It's under half. So the spectrum of the PMT proteomics is being overwhelmed by large proteins. We have to get rid of those large proteins, or you're not going to see these smaller proteins. It becomes a matter of probability at that point. And this is what our lab does. So we borrowed from biochemical extraction techniques to find neuropeptides from Wiley Dale's lab and Roger Gielin's lab. And their staff, especially Wiley staff, is out in our lab. They've been in the lab for like 50 years. It's actually pretty cool to see how things have progressed. They discovered somatostatin, CRH, TRH, and more. And Joan Vaughn, who's in Wiley Dale's lab, she's the one who does all this biochemical extraction. And so we use those biochemical extraction techniques to find these microproteins. And to appreciate generally what this looks like is if you take a Western blot, or if you just run this on an SDS page gel, a protein extract, you'll notice that the majority of proteins migrate above 70 kilodalton. Now, if you apply these biochemical extraction techniques, such as acid extraction, which is precipitate out larger proteins, as well as a smaller carbon chain column, we can flip that. We now have a tissue extract that's about 90% enriched with proteins under 70 kilodalton. Then at this point, we can just go even deeper and start fractionating out. That way, when we shoot this into a mass spec machine, we increase the probability of finding those smaller proteins that otherwise would be masked by the more abundant larger proteins. And here's an example of one. So here's a microprotein that's has substantial missing data in the ROS-map cohorts. However, even with the missing data, there's a hint of it being highly expressed in Alzheimer's disease, but about a third or almost half of the data is missing from the proteomics experiment. Now, if you go to any type of tissue repositories, in my experience, I can only get 100 milligrams of tissue. That's what they're willing to get out. They got to get out other tissue, other people. That's all you really got to work with. But I need more protein, effectively saying. That's where our extraction techniques come into place. So we've developed a radioimmunoassay, an antiserum against this microprotein, and at 100 starting milligrams of tissue, you can't detect this thing, even with a highly sensitive radioimmunoassay. Now, if you apply our small peptide enrichments, then you can start quantifying this. So I can take smaller tissues now, because of this extraction technique, filter off the larger proteins and increase the chance of finding and detecting and quantifying these proteins at the peptide level and match that to a lot of the transcript level. And you can see this highlighted here in green. If you get 250 milligrams to start with, you're going to quantify this a lot. And so we are now doing this at ROS-map in collaboration. So we have a pilot sample of several dozen brains where we're going to apply a lot of these biochemical extraction techniques, overlay that with our computationally defined search base of small proteins. And the hope is by next year, we'll have a quantifiable starting database where we can disperse to everyone in the AD knowledge portal that you can implement into whatever pipeline that you have. And these should represent either smaller protein isoforms of known proteins, or they should represent new proteins that are misnomers as non-coding, as well as maybe some of these pseudo-ors that otherwise you can't detect at the transcript level. And that's the goal. And hopefully that happens by the start of 2026. Back to the very last slide. So how do we address this starting point in 2004? We need to go deeper and extract and isolate these peptides. Borrow the same techniques that found CRH and somatostatin, etc. We need to have good transcriptomes. So the fact that we have a long read transcriptome, we can get some of those unique exons. We also need to accelerate functional studies, which is where the omics feature comes in. If there's an association at the genetic level or the transcriptome level with the disease, where we can target those to those specific disease relevances, and also speeding this up. And that's hopefully what we're going to try to do really fast, because the faster we go with this, and the faster we can train better models and reduce a lot of the false positives that naturally we're going to have with the field that's just starting out. So I'd like to thank all of our funding, the Salk Institute, Aleksandr Tyshkovskii, my current PI, Joan Vaughn, who's all biochemistry, several postdocs, USC, former lab, Hossie, Eileen, Arts, Rush, we have Bennett, Adney, who helps with a lot of our genomics, and our funding NIH, and the Clayton Laboratory for Technology. All right, thank you.
Any questions for Brendan? We are about to go into our afternoon coffee break. So if you had a question or a longer comment, you can also chat with him during the break.
Hello, everyone. I think we're gonna start with the next session. We're getting ready towards the end. Thank you for staying around. So we have 3 more sessions. The last one is gonna be over Zoom. So just hang in there. So this first one is by Katrina. They're coming from the East Coast, actually. It's a good time, I guess, to begin the presentation. Everyone has around like 20 minutes.
No.
You're on mute.
real loud, wouldn't you?
Is this on?
Yeah, there it is.
Kind of project.
That was just you.
Yeah. it was just-
He projects very
I'm
well.
kind of loud.
Right, I don't think I'm gonna take all 20 minutes, we'll see how this goes. This paper is looking at sex differences and the relationship between APOE4, cognitive decline and dementia onset. It's collaborative projects with my PhD student, Jennifer Alshire and Eileen Crimmins here, and it's supported by funding from the NIA. Okay, so, oops, I'm doing it wrong. Okay, so we're kind of switching gears because we're not talking about epigenetics at this point, but we're talking about APOE4, which we know is a strong genetic risk factor for late onset Alzheimer's disease. It results in about a 3 to 15 times increase in dementia Alzheimer's disease if you have one of these E4 alleles. And so one of the things that we found in the scientific literature was that there's a lot of discussion around this idea that women who are APOE4 carriers tend to have like a stronger effect, as a higher risk of cognitive decline and dementia onset if they are one of these carriers. This is it's not always consistently found, but it is found in a lot of these clinical studies. And I just have a few of them highlighted here to show you the different types of cognitive outcomes that have been examined and the different types of samples that have been used. And what you might notice is that these studies are all volunteer studies. They don't, I should have prefaced this by, I'm a population researcher. And so my focus is really on the social aspect. I do a lot of work on population-based studies, but what I mean by population-based studies are probability samples. And so I'm really interested in making sure that samples are representative of the population that we're trying to make inferences of. And so looking at this body of research and seeing that a lot of the samples are drawn from convenience samples, volunteer samples that have unknown bias was something that really kind of piqued my interest. And I was really curious whether we would see the same effect if we were to look in a sample that was drawn using probability methods. So that's really what the study is. We wanted to test for sex differences and the relationship between APOE4 and cognitive decline and dementia onset in a sample where the men and women were representative of the entire US population of non-Hispanic Black and white midlife and older adults. And so we use a health and retirement study. I feel like some folks might have talked about this earlier, but I'll just briefly discuss the health and retirement study in the sense that it is a probability sample. So it's drawn using probability sampling methods. This is a biennial survey of US adults age 50 and over. It started in 1992. Data is collected every other year. People are followed over time. There are replenishment cohorts. So every 6 years, new cohorts who are 51 to 56 years old enter into the HRS. And so it always retains its representativeness of midlife and older adults. In 2006, they started a genetic sample of community dwelling adults. And these folks were followed if they did become institutionalized, but they were in the community at the time that their blood was drawn. And this was collected in 2006, 2008, 2010, and 2012. And then the cognition and dementia measures were assessed every 2 years. We use the data from 2006 to 2018 waves. And so you can see in that kind of, I don't know, decision tree or whatever you want to call it, how we broke down our samples. So we restricted our sample to people who participated in the genetic sample collection. And then we restricted our sample to folks who self-identified as non-Hispanic, white, or Black. And then we further restricted it to people whose PC loadings indicated that they were either of European or African genetic ancestry. And then for the cognitive decline analysis, we restricted it to those born before 1960 so that they had sufficient numbers of waves of observation to look at decline, and had at least one measure of cognitive function between 2006 and 2018. You can see that we ended up with about 11,000 white adults and about 3,000 Black adults. And so that's our sample for our cognitive decline analysis. Then for our sample for dementia onset, we restricted our sample to those who were born between 1924 and 1959. They couldn't have had dementia before or been, indicated dementia before age 65 or baseline, and they had to have been observed after age 65. So that resulted in about 8,300 white adults and about 1,500 Black adults. Okay. It looks good up there, but it does not look good here. Anyways, so I just wanted to show you what the frequency of APOE4 allele frequency was in the sample for white adults. For men and women, it was about 26%. This is at least one, so we collapsed 1 and 2 together because there wasn't sufficient numbers of those with 2 E4 alleles. There wasn't a statistical difference in allele frequency of sex. For Black adults, it was about 40%, which is consistent with the literature. And again, there's no statistical difference by sex. All right, for cognitive function, we use the 27-item modified telephone interview for cognitive status. This is a validated measure of cognitive function. It consists of items on immediate word recall, delayed word recall, serial sevens, and backward counting. And you can see the distribution of the cognitive function scores at baseline here. I plotted it for women in purple and men in aqua, for white and Black mid and older adults, and for carriers and non-carriers. And you can see that the distribution is quite consistent. There doesn't seem to be a large sex differences across any of these different groups. Okay, so for dementia status, we use the Lange-Weir classification with validated cut points for those who use the self-reports, the TICS item, or for those who have proxy scores, so proxy or interview assessment coupled with their score on IADL. And then here you can see the white respondents and the Black respondents, that those who were E4 carriers had higher risk of dementia onset, and both for white adults. We didn't see that effect for Black adults. We also saw in these bivariate analysis that there was a gender difference between dementia onset between men and women and white E4 carriers. So just want to note that. But this is unadjusted models. Okay, so our analytic approach was to run race stratified models. And then for the cognitive decline, we use linear mixed models. And we included an age and an age squared to fit the functional form. We interacted our variables with age to assess rate of decline. We included SX by APOE4 interaction term to determine whether there were differences in the relationship between APOE4 and cognitive decline. And then we also adjusted for birth cohort, interview mode, age, the principal components for population stratification. And I forgot to add here also years of schooling. So that is also a covariate that we included. And then for our dementia onset models, we ran Cox proportional hazard models, and we determined age at onset as a midpoint between the last non-demented assessment and the first observed with dementia. And then those who left the study without dementia or did not have dementia by the last observation were considered censored. Okay, so this is basically our findings. We didn't find any APOE4 by sex differences in level or decline in cognitive function among either the white or the Black HRS participants. You can see the women are in the kind of purple lines and the men are in the aqua lines, and there's really no difference in their rate of decline. There's definitely no statistical difference. Okay, and then for dementia onset, we basically found the same thing, that there was no APOE4 by sex difference in transition to dementia among either the white or the black ADRD respondents. Now, then did a bunch of other types of sensitivity checks because we wanted to see whether these would be robust if we considered other alternative specifications. So for instance, we wanted to account for selection bias, healthy survivor bias, and we wanted to consider the fact that in our original models, we also included people with a E2, E4 genotype, and we know that there's some research that suggests that E2 is protective against dementia, so we wanted, as others in the clinical field have done sometimes, not always, but remove them from the analysis and see what happens. So in the clinical samples, a lot of these samples are really highly educated, they live in urban areas, so we wanted to rerun these models in a similar type of sample. So we restricted these to people who had 13 years or more of schooling. You can see there. I highlighted the gender by APOE4 interaction to show you what the effect is on the level. So that's at age 70, that's the first column, and the rate of change in cognitive decline, which is the second column. And if it's statistically significant, you'll see a little asterisk, and you can see across the whole different types of specification, there is only one. And since we ran like 50 models after adjustment for multiple comparisons, that's not significant. It was actually almost not significant in this model. It was like 0.049. So just to be frank. Okay, so what we found was, restricted to high educated folks, no effect. Urban folks, no effect. In the mortality analysis, what we did was we gave everybody the value of 6, which is the threshold for classification of dementia on the ticks. So we gave them that value of 6 in the for every year or the year before, the year between the last observed and death, to basically say, okay, what if folks who die had dementia before death? So it's a very conservative test of this mortality effect. And then we wanted to say, well, okay, what if we restricted this to people who are younger than 75 so that we can maybe take care of potential selection into older ages, right? So we did that, no effect. The only time we found any potential signal of effect is when we excluded those E2, E4 genotypes. So like I said, it was a 0.049 statistical, a p-value, and we did a bunch of comparisons. It's really not significant. We did the same thing for the Black participants, and here you can see again across the board, no effect. It's no matter what we did. I educated urban residents, accounting for mortality, looking at younger ages, and even excluding the E2, E4 genotypes. Then we move on to the dementia onset, where we did the same thing. We restricted it to the high educated, those who lived in urban areas, the younger respondents, and we excluded the E2, E4s. Again, we don't end up seeing an interaction with sex across any of these models in the white respondents. And we also don't see that in the Black respondents either. Okay, so in conclusion, we used a nationally representative sample of white and Black older adults, and in that sample, we found no evidence that the relationship between APOE4 cognitive decline and dementia onset differed by sex. These findings, I think, challenge prior research that are based on clinical and community samples, which are often volunteer convenient samples, and that often find stronger effects of APOE4 in women. But because those studies rely on highly selective, non-probability samples that are more educated and urban, we tested whether restricting our sample to similar groups would produce the same kinds of patterns, and we didn't find anything. Our results suggest that previous reports of sex differences may be due to sex differential participation bias in clinical studies, meaning that there are unmeasured differences in why men and women participate in clinical studies rather than actual differences in genetic risk. There's also some indication that maybe researchers define inclusion and exclusion criteria, such as how we treat the E2, E4 genotype may also contribute to some of these inconsistent findings. Overall, our study underscores the importance of using probability samples to better understand genetic risks of dementia. I'm going to plug that as a population scientist. And we think, though, of course, that this is one of the only studies we found that actually use a probability sample. So we need to replicate this in other studies that also use probability samples in diverse, nationally representative, and even international samples to really understand whether these results are robust. So that's it.
Any questions? Oliver? It's a little bit general. I mean, I have a student who is doing exactly the same thing in a terribly clinical study. She does find some sort of, where she's looking at sex differences, and that seems to be magnified among areas. But obviously, there's probably a lot of bias that we're ignoring. But we're always debating. She's always telling me that dementia is higher in women, and is it just age? And you must have looked at this in HRS.
I just turned off my mic. I'm gonna pull an Eileen and say, is it? Is dementia really higher in women? I think that, like, if you think about it in terms of population and mortality and who dies at younger ages, and it's hard to know whether the reason why you're seeing women with higher rates of dementia at older ages is because they actually survive at older ages and then die. So, I mean, I don't know that that has anything to do with sex differences in, like, dementia, other than social differences in who dies at what ages, right?
There are also cognitive impacts of caregiving for other people with chronic illness like dementia. So if your husband had dementia and you took care of him and you lived later, and you also might have the cognitive impacts of the self-neglect that happens when you're doing chronic caregiving, like you have to tease all that out. Like, what is it that makes older women more vulnerable?
Yeah, I mean, I think that goes to the social aspect of, of what, you know, how women experience some of these things as they age.
Yeah.
But you're saying they are more vulnerable.
Well, I mean, they live longer, so they're more vulnerable. They do more caregiving, so they're more vulnerable. There are other ways that the world affects them in a deleterious way that might make them more vulnerable. But are they genetically more vulnerable? I think what she's saying is, for this particular marker, no, that's not it.
I mean, it was an unfair question. It's not really.
Oh, it's like
It's a key thing that I can develop.
It was just a question.
A quick question. So I might be understanding the graphs that you showed, not the graphs, sorry, the tables that you showed incorrectly looking at cognitive trajectories. But in that one, were women consistently doing better at baseline in your sample?
Yeah, so this goes back to the question. This is cognitive function. So yes, like at age 70, women had higher cognitive function scores than men did. Their rate of decline was no different, it looks like, at least in the highly educated. I'm just looking at what I have here on the tables. Yeah, those the rate of change. I know I kind of graded out so you wouldn't look at all of the main effects, but like that main effect on the top is really the gender effect. And so yes, they had higher rates and no difference in rate of decline.
Why do you think you're seeing that? Do you have a hypothesis about that?
I mean, I think that I feel like this is a pretty consistent finding in the literature when you're looking at population, when you're looking at probability samples in terms of women perform better on cognitive function measures than men do.
Certain ones, but this is a text of the feeling.
It's a text.
Yeah,
It's
interesting.
a combination of word and numbers.
Mhm.
And I will tell you, if you separate it into words and numbers, you get sex differences that are different.
That one's
If you just
words, yeah.
put it together, this is what you get.
Oh, sorry, in the back.
Yeah, thank you. This was great. I'm sorry if I missed it. I know the trajectories you showed were nonlinear. Did you allow for nonlinearities in age for the interaction term as well?
We tested it, it wasn't significant. So we just simplified the model.
Okay. And did you use the sampling weights in the mixed specs
Yes.
models?
Sorry, I should have clarified that, yes, for sure, because like without the sample weights, then you're not adjusting for this, the fact that it's a
Right.
complex sampling designed to plot different probabilities of selection. So yes.
Were you thinking we should?
No.
Oh,
It's just, yeah.
Any other questions? Thank you.
All right, great. I'm Sean Curran. I'm a professor here at the Leonard Davis School of Gerontology. I'm a biochemist by training, but my lab studies both molecular biology and genetics. And so we've heard a lot about sort of this risk spectrum for ADRD. Most of what we know about the etiology of Alzheimer's disease comes from just 3 dominant proteins, PSEN1, PSEN2, and APP. We've heard about APOE, which I'm gonna talk a little bit about today too. But the point of this slide is that really we need to rapidly discover other components that are going to lead to this disease so that we can interrogate them, both in the laboratory as well as in the clinic. And my talk is designed to really lead you on a journey from how I started in this small nematode, C. elegans. Don't worry, these are not to scale. Use that as a really powerful genetic model. But we've translated that into both studies in mice and with the help of Em and Eileen into humans as well. And so regardless of what you think about the hallmarks or the pillars or however we're describing these components of aging, a very clear unifying theme across them is that homeostatic disruption of any one of these pillars is a common feature across any aged person. And so my lab focuses on one transcription factor, these proteins that go into your nucleus, turn on genes when you need them. That factor is SKIN1 in that nematode. It's NRF2 in you and I. And all I want you to appreciate from this slide is that this one transcription factor plays multiple roles in the cell. It's involved in innate immunity. It's canonical roles in ROS and redox balance, but it balances proteins when you need to make them and when you need to turn them over, changes metabolism. And it does this differentially across all of your cells, really responding contemporaneously to the needs of the cell in that one given moment. And so we look for ways to rapidly screen for genetic factors that will lead to increased resistance of stress that happens just normally through life. You can see the types of stress that we look at here, oxidative stress, just if you cut an apple in half. You cover one part of it in saran wrap, you leave the other part open, you see the part that browns. So that's a really easy example to understand oxidizing stress. We fortunately have more sophisticated reporters that we use in the lab. Here, what we're using is actually a sensor for when that transcription factor is turned on. Transcription factors bind to promoters. Promoters then are the front part of a gene that allow the gene to be transcribed. Here, we're using a very strong reporter for SKIN1 activity. It's called GST4. You don't have to remember that for the rest of my talk. But when SKIN1 is activated, these worms will glow green when you shine blue light on them. What I want you to appreciate is that in this picture here, you don't see any green. And that's intentional because we actually, all of our cells, every organism tightly regulates the activity of SKIN1 to make sure that it's off when you don't need it and only gets turned on when you do. So when we do genetic screens, we're looking for things that look like this. These bright, screaming, fluorescently green animals that we know now have activated SKIN1, even though they're not undergoing any types of stress. So this animal thinks it's under stress attack, has activated SKIN1 inappropriately. Now, what you maybe would expect, if we challenge that animal with acute stress, either giving it paraquat, exposing it to air pollution that the Finch lab collects from the 105 freeway, give it exposure to a pathogen, it does so much better than animals that don't have activated SKIN1. You can appreciate that from that middle graph there. These are animals that are dosed with a really high amount of the oxidizing agent paraquat. 90% of a wild type animal will die from this acute treatment. Nearly 100% of the animals that have this pathway activated completely survive. Now, we were excited about this. This discovery came about 15 years ago when I first started my lab. We said, wow, this is going to be a really long-lived worm. It's going to, these worms only live about 30 days. We thought, wow, this is going to live for months. It's going to be incredible. Genetics is really humbling because it can immediately disprove your hypothesis. And sometimes that's fun because you get to figure out why you were wrong. But really, none of the animals that we identified from these screens are actually long-lived. In fact, most of them are exceptionally short-lived. And that goes across all of the different types of mutants that we've identified in the screens throughout 15 years of work here. So what this really told us is that turning off stress responses is probably equally as important as being able to turn them on. And as much as I know you guys all wanted to hear a talk completely about C. elegans, I'm going to disappoint you. This one gene that I'm going to tell you about today is called WDR23. It's a negative regulator of SKIN1. You get rid of it, SKIN1 becomes constitutively activated. There's almost a perfect isoform in mammals, in mice, as well as in humans, that have essentially the same structure. And theoretically, we tested whether or not it would regulate NRF2 in people and in mammals, just like it did in C. elegans. So we took the mammalian version of this gene, we expressed it in cells to see where it localizes. It ends up being one isoforms in the nucleus, one isoforms in the cytoplasm. NRF2 works in both the cytoplasm as well as the nucleus. It regulates NRF2 in both of these compartments. All I'm showing you here is that if we overexpress, introduce extra copies of this WDR23 gene into these cells, it reduces the amount of transcription that NRF2 does. So really, it's also a regulator of NRF2, just like it is in C. elegans. This was actually surprising because decades of work defined a completely separate system for regulating NRF2, and it's called KEEP1. And so really what this initial work really described for us is that the regulation of NRF2 is even more complicated than we initially thought. There are two independent mechanisms that function in parallel to control the activity of this transcription factor. This is enough for me to say, okay, well, maybe we should keep focusing on what WDR23 does for NRF2 in mammals. So we generated a knockout mouse where we removed the only copy of WDR23 to see what its effect on physiology was. So we've tested multiple tissues, we've looked at metabolism, we've looked at reproduction, but some of the most interesting findings that we found was when we looked at what WDR23 is doing in the mammalian brain. And so the leftmost picture is a Western blot showing you how much NRF2 protein is in these animals. You can appreciate that that band that's being pointed to by the arrow that says NRF2, there's more of it in animals that don't have WDR23. So in the brain, if you don't have WDR23, you have extra NRF2 that's acting there. The right blot is RNA-seq analysis of hippocampal sections of these brains. There actually wasn't a lot of increased transcription, but about half of the gene targets that we saw that were increased have canonical NRF2 binding sites in them, telling us that the activity of NRF2, not just the levels, are increased in these animals that have lost WDR23. We look at what types of genes are being regulated by NRF2 in the brain in animals that don't have WDR23. Some of the larger hits that come out of these Go analyses are things that are involved in cognition, learning and memory, spatial awareness. And these are maintained both early in life, in midlife, as well as in relatively old animals at 24 months. And so because of this, we crossed that animal that doesn't have WDR23 to probably what's the most well-studied model for AD, at least in the mouse community, 5 or 6 years ago. This is triple transgenic model. This is an exceptionally accelerated model for Alzheimer's disease and dementia in a mouse. It's expressing all three dominant forms, dominant alleles that have been identified in humans that drive AD pathology. And so it's pretty much a triple whammy of this animal. They get dementia very quickly, but it's really how we learn the etiology of the disease, what comes first, right? So amyloid plaques, then tau fibrils that lead to impaired cognition. And so in multiple tests that we did in these animals, comparing that triple transgenic model to that same model that's now missing WDR23, the animals missing WDR23 actually did significantly better in all of the tests that we put them through, both in their activity, both in their reduced anxiety in an open field test, their ability to maintain cognitive function by doing this alternating behavior in this Y maze, where they have to go to all three compartments after each other without repeating. that's normally lost pretty quickly in the triple transgenic model. It's maintained quite well in these animals that are missing WDR23. So in general, I would say that an animal that's missing WDR23 that has more NRF2 does better in this rapid AD model than animals that have normal function. What was super surprising from this is that when we then looked at the pathology in the brain of these triple transgenic animals, there's absolutely no difference in tau tangles. There was no difference in the amount of amyloid that was being accumulated in the sections of the hippocampus. That told us that any cognitive improvement that was happening from the loss of WDR23 and the increase in NRF2 is independent of the amyloid hypothesis. In fact, I don't know that it actually ever reached statistical significance, but if you just look at the pictures here, those WDR23 animals have more amyloid in them and actually have worse tau tangles, which I'm not showing you here. But so really this suggested that this neuroprotection that's coming here is independent of this pathway that had been most well studied. In fact, if we look go back to that transcriptional analysis that we did of the hippocampus, but now in the triple transgenic model, NRF2 does show up as one of the transcription factors that's being activated in an age-dependent manner in these animals. But there's quite a few other transcription factors that are doing even more in the hippocampus of these animals. In fact, most of these regulate metabolism and not oxidative stress resistance or proteostasis. In fact, one of the most strongly upregulated genes that we find are both in lipid metabolism as well as in glucose handling that's happening in the brain of these animals. And so this was a puzzle, and we wanted to figure out, how can we sort of narrow down of all the possibilities of what WDR23 is doing in the aging hippocampus, either just in a normal setting or in the context of some of these dominant drivers of AD pathology. And we turned to a core that Eileen and Em put together as part of our Nathan Schock Center, which really is a discovery engine that folks like me, who don't work in humans, but work in model systems, can leverage to take data that we've already acquired in those models and see whether or not there are any genetic variants that might be in the HRS or in ELSA or in any of these other population-based studies that might tell us what's going on in the context of natural human aging. And so we presented this data to Em, and I showed her, okay, look, WDR23 seems to be linked to increased performance. It maybe has an effect on dementia. It seems to be independent of what's happening with amyloid or APP. How do we figure out what's going on in here? And so she took that data, she queried in this GeneWAS that they put together to look at any SNPs in human WDR23 to see if they correlate with any of the measures of dementia that are in the HRS. And it turns out humans have interesting biology too. There's actually at least 3 SNPs. Actually, one's not coming up. It's light green on the bottom too. But there's 3 SNPs that are associated with WDR23 in humans that seem to have a protective role, at least in the populations that we were able to query from the HRS data sets. But what's super interesting is that they only provide that protective effect in the context of the APOE4 allele. And the ones that we were looking at here, we excluded APOE4-2 hets, because that isn't necessarily, as we just heard in the previous talk, that isn't necessarily a risk factor, but they had at least one copy of the APOE4 allele. And so really, I think this gives us sort of a tantalizing idea to follow up now that WDR23, I think we have really strong data that it does play a role in cognition, has a role in maybe natural brain aging, but not in the most well-studied dominant drivers of Alzheimer's disease for at least based on the amyloid hypothesis, but perhaps in relation to whether or not you are more likely to get dementia if you're carrying one of these APOE4 alleles. And so the things I sort of want you to take home from this talk, one, leverage appropriate models to accelerate discovery. The screens that were done in C. elegans were done all by by graduate students. They take about a month and a half to complete. They take about 3 to 6 months to identify the causal mutation. And with tools now that we have in this geroscience realm, you can rapidly translate discoveries like that into a human-based system pretty quickly. If anything, it generates new hypotheses that you can then test in those models again. And I think this is perhaps one of the strong points of this geroscience idea. You can leverage multiple models, you can accelerate discoveries much more quickly, and you can test things if you use the appropriate model. I think identifying additional genetic variants like this, ways that we can do both gene by gene analysis as well as gene and environmental effects, I think are really easily testable both either in an invertebrate or a mammalian system. But really, we've heard this a lot today already, we really need to look beyond these A-beta models and these simple aggregation ideas as being these drivers of Alzheimer's disease and other dementias. It's not that they're unimportant, but I think we've spent enough time and effort on them that I think it's, let's see what else is out there and see, maybe it leads us back, but we'll see. But really the ultimate idea here is that we can use a precision-based approach, either informed by the geroscience idea, but take a person's individual variability, what their environment is, in order to better prescribe an approach to either alleviate the conditions that are going to come naturally with age, or combat an age-related disease that a person might be predisposed to. These are all the folks that did the work. Specific to this project are the ones that are highlighted in red. I'll thank our funders, and I'm happy to answer any questions that you might have.
Any questions?
So in human studies, you tend to see that individuals with elevated amyloid beta and tau that are still pretty well cognitively preserved tend to have better synaptic density. And I was just curious if you looked at synaptic markers in your rodent models in the hippocampus.
We haven't yet, so it's a great question. But synaptic function, at least in the older animals that we queried, so the 2-year-old mice, maintenance of synaptic plasticity was one of the top ranked Go term sets that are in there. So being able to map that, I think, would be a great idea.
Any other questions? Oh.
We're good.
Hi, everyone. I'm sorry that I am not there in person to present to you today. You can blame this guy. This is my son who had a skiing accident on Monday. So we have been meeting with various surgeons and getting lots of scans and x-rays. So that is why I'm not with you. He's fine though. I think it's just an elaborate ruse to get out of school and have me serve him. But whatever. Today what I'm going to talk about is epigenetic G. So we're going to turn back a little bit to talking about epigenetics. But like Sean just said, I think the theme here is still the same, which is when we think about sort of cognition and our ability to predict dementia in population-based studies, kind of thinking broadly. This is work collaboratively with Matteo Farina and Coulter Mitchell, who are with you, but they haven't seen these results, so they can't answer any questions quite yet. So any inaccuracies are my own. So just a little bit about epigenetic G. This is really an epigenetic marker of general cognitive ability, or G. And it was developed, I'll talk about it more, using an epigenome-wide analysis of cognitive abilities. And here we're going to take that and leverage it in the health and retirement study and see what we can tell about the relationship between epigenetic G and education, cognition, and how well it can predict dementia on its own and with other neurobiomarkers that we have. So the background here is that this was developed in the Generation Scotland study. And really what they wanted to do was create an epigenetic-based measure of cognition, in part because not all studies have measures of cognition, and they wanted to see how well they could do with methylation. And they wanted to see how well this worked in addition to kind of more traditional genetic markers or polygenic scores that they also had in this study. So they develop it in Generation Scotland and then test it in the Lothian cohorts. So again, G is this general cognitive ability but test. And so it's a predictor of intelligence and cognitive function. And it's really derived from multiple cognitive tests. Again, they use participants in the Generation Scotland study. So a subset of them, about 9,000, have methylation measured. They range in age from 18 to 93, so covering the full adult age span. And like I said, the out testing was done in the Lothian birth cohorts. So the phenotype to create G is really a combination of a lot of different cognitive tests. So it has elements of memory and also digit symbol test, substitution tests, verbal fluency, and vocabulary. So this G is calculated from the principal component of those tests. And then they looked at all the CPG sites that they had measured. In this case, it was the 450K. They found 3 CPG sites that were strongly linked to cognitive function in the EWAS analysis. But then they also develop an epigenetic score they call an EPI score. And this is just the weighted sum of all those CPG sites that they looked at. So all of them, not just the 3. And its relationship to predicted G, so the general cognition measure that they had. And then they compare this to the polygenic score of cognitive ability that they also have measured in this study. And essentially what they found was that methylation explains some variation in G. And again, those 3 specific CPG sites were significantly linked to cognition. In the out testing samples, their epigenetic score predicted somewhere between 3.5% to 4.5% of the variance in G. And that wasn't as much as the polygenic scores they had for cognitive function, but together they sort of summed so that together the polygenic score and the epigenetic score explained 10%. So it's adding something new. It's not the same as the genetic marker. And they also showed that this epigenetic score correlated with brain and inflammatory markers, so brain volume from MRIs and inflammation-related proteins. They looked at, so here kind of the orangey pink is the epigenetic score and blue is the actual measured G. And they kind of saw that there were the same relationships between epigenetic G and measured G and all sorts of things that we look at related to aging. So lung function, hypertension, grip strength. It sort of did a similar job in its associations with those things. They also had really fancy MRIs, so they looked at how similar the epigenetic score and actual measured G were to regions in the brain, and largely they overlapped. So the pink is where the different areas that were associated with these markers overlap. Anyway, they felt pretty good about their marker of epigenetic G. So where this has been used in other population studies, it's pretty limited so far. There is a study by Laurel Raffington that used epigenetic G from buccal DNA methylation in a German cohort, again, ranging in age, but mostly older adults. Looking at how it related to socioeconomic status in terms of household income and highest education level in the household, how it related to age. And essentially what they found was that epigenetic G had sort of an expected age-related trends, so higher in younger adults and stabilizing in middle age. And in their study, SES was not significantly associated with epigenetic G, so education in this case. So the highest education in the household wasn't associated with epigenetic G. But we wanted to see how this looked in the health and retirement study. So what we sought to do was examine the relationship between epigenetic G and educational attainment, parents' education, cognitive function, and dementia risk. We also wanted to see how epigenetic G related to the educational attainment polygenic score and also the general cognitive function polygenic score from the Davies et al. GWAS. And we wanted to see how well epigenetic G did in predicting dementia compared to other neurodegenerative markers and genetic markers. So we used the health and retirement study, which Katrina already talked about. We median imputed any missing CPGs from our DNA methylation measurement. And we had 97% of the CPG sites that were in that generation Scotland EWAS. So we were able to create what we felt was a pretty good version of their epigenetic G score. We constructed it for all 4,000 and some people from the HRS 2016 venous blood study participants who have DNA methylation measured. As Katrina already talked about, we looked at the cognitive tests in the HRS. So this is immediate and delayed word recall, serial sevens, and backwards counting. So like G, it's sort of a mix of different skills. We also use the LingoWeir diagnostic algorithm to define dementia status and use the appropriate sample weights. So what we saw was that epigenetic G was associated with education. So in exactly the way you would think. So you have lower epigenetic G if you have lower levels of education and higher if you have higher. We saw an age-related gradient such that people who were older had higher epigenetic G. We saw some differences in sex. So females generally having lower epigenetic G and differences in race and ethnicity as well. So with our white, non-Hispanic other group having the highest scores and our non-Hispanic Black and Hispanic groups having lower scores. This is just to show the relationship between epigenetic G and education and in regressions. And you can see that it's exactly the way you would expect, which is lower epigenetic G with lower education. And we see the same thing with parents' education. Probably the easiest one to look at is the table on the bottom here, where you see the parents' education. We have no parent has education greater than 8 years. One parent has greater than 8 years of education, or both parents have greater than 8 years of education. And again, we see lower epigenetic G with people whose parents have less than 8 years of education. But it's very clear that this is measuring something different than the other neural markers that we have. So on the left here, you can see NFL, GFAP, the A beta 42 and 40 ratio, and p-tau 181. These are all neurodegenerative markers, some AD specific, some not. And they really are not correlated at all with epigenetic G. We also looked at how well epigenetic G was the correlation between epigenetic G and our polygenic scores. Here we do it separately by ancestry because they are different in different ancestry groups. And again, we don't see a very high correlation between educational attainment or the general cognition polygenic score and our measure of epigenetic G. So clearly we're getting at something different here. And I'm going to apologize for all of these really big tables, but I will just narrate as we go through. These are sort of hot off the presses this week. The first thing we looked at was, does this even relate to cognition in the HRS? So this is the participants' cognitive score in 2016, the year that we took the DNA methylation measurement. And epigenetic G is a statistically significant predictor of cognition at the time of methylation collection. And here, as we move through the models, we adjust for age, sex, race, ethnicity, and education. And as we adjust for all of those things, epigenetic G remains a significant predictor of cognition. But interestingly, it's an even stronger predictor of baseline cognition. So the earliest cognitive measure we have among our participants. So the beta parameters here are a lot bigger. And again, it's robust to us adjusting for demographics and education. When we look at incident dementia, again, we see that epigenetic G is protective. These are not odds ratios, but they are just the parameter estimates. But epigenetic G is, again, statistically significantly protective of incident dementia, no matter whether we adjust for socio-demographics and education. We wanted to see whether it would still hold up in models where we've added the neurodegenerative markers. So here we add the ones I talked about, NFL, GFAP, A-beta 4240, and p-tau 181. And again, epigenetic G remains a significant predictor of incident dementia. It's a protective effect. And also when we adjust for APOE, either having one allele, E4 allele, or two. And again, here, when we add the educational attainment polygenic score and our general cognition polygenic score, epigenetic G maintains its effect, even when we adjust for those things. Here, we're looking at the European ancestry group, but the same is true among African ancestry group as well. Again, still a significant predictor, even when we adjust for the neuromarkers and genetics. Among Hispanics, the story is a little bit different and we don't quite understand yet why. So it's still a significant predictor, but now it's no longer protective. There's a slight risk element of having a higher epigenetic G among Hispanics. So I don't have like a pithy conclusion here, but clearly there's something here that is really interesting and different than the other types of markers that we have. So our plan is to explore looking at it more, and especially looking at differences in the race and ethnic groups in terms of its ability to predict outcomes. That's all I have. Thanks.
Oh, yeah, it comes pick up here.
So Jessica, it's
Yep.
Katrina.
Hi, Katrina.
So, coefficients, can you explain, because some of them look really large, is your EpiG a standardized measure? So that's like a unit, like a standard deviation is related to a change?
Yeah, no, we talked about it today to make them standardized, but they're not. These models don't have them yet standardized. So they are, and again, those aren't odds ratios. Those are the parameter estimates for incident dementia. In terms of odds ratio, it's like I think the fully adjusted model, it's like 0.2.
But what about the cognitive function estimates?
Yep. Yeah, so the scale of epigenetic G is a little bit wonky. So I think in our next version, we're going to go through and standardize everything so that it's a little bit easier to interpret.
Hey Jessica, it's Eric. So I noticed that your associations got way stronger after controlling for age, sex, and race ethnicity.
Yeah.
Have you tried looking at the associations with the cognitive biomarkers after controlling for age, or was it just those correlations?
No, so here, this one is, if you go from here and you see the estimates about -15, and then now we adjust for the neurodegenerative markers, it stays the same. Is that what you're asking?
Right. I meant with the, did you ever try the neurodegenerative biomarkers as the outcome in these regressions?
No, not yet.
So that would be interesting, I think.
Yep.
Hi, Jessica. Do you have just what is the straight correlation or R squared of G with the cognitive measure in HRS?
No, not yet.
Jessica, you do have markers of information processing speed in HRS, do you? So reaction
not
time?
directly. It's something that we can get in a really rough way from the keystroke data, so the timing data as they go through the survey, especially in like the self-administered internet panel. But that's probably like the only one, or maybe like animal fluency in terms of how fast people are able to get things done, but we really don't have a straight measure of it.
Even in HCAP?
Well, in HCAP we might, but not in the core. and not for more than one wave right now. What are you getting at, Cathal?
Well, because what the argument went at least 20 years ago that the underlying genetic architecture for G could be related to the speed of differences in speed of information processing, which may also be related to the degree of myelination of cerebral nerve fibers. So I just wonder if you would look first at the relationship of EpiG with your speed of information processing measure, and then you look to see whether the relationship's attenuated when you adjust for your measured speed of information processing. Yeah,
I mean, so we have a lot of overlap with the HCAP sample and the methylation sample, so we could definitely do that, and we could look a little more closely at the relationship with the different types of cognitive tests that we have instead of just lumping everything all together. So it's a good suggestion. Thanks.
Any other questions, comments?
I guess, hey, it's Jen. Hi, Jessica.
Hi.
How are you? I was just trying to think what an epigenetic marker of, like what an epigenetic, like what's affecting these epigenetic markers over the life course potentially. And I guess obviously cognitive stimulation, educational attainment might be the causal. I guess there's some idea of what some kind of cognitive exposures are creating these markers. And so it did look like there was a big drop once you adjusted for educational attainment in one of them. But I wondered if that might explain some of the differences across race, ethnicity, that there were just kind of different exposures, you know, sculpting the brain in those groups because educational attainment wasn't as available, or something along those lines, or what you think, yeah, the life course kind of exposures in theory would be that contribute to these markers.
Yeah, I mean, I think that in much the same way that we think about the relationship, kind of the social and environmental relationship between these exposures and cognitive test performance itself, I think this is sort of capturing a similar set of things. So, you know, you can think about it not just like years of education, but quality of education and maybe cognitive stimulus through occupation. You know, and obviously those things aren't evenly distributed in our country. So we don't see them evenly distributed across race and ethnicity here. So that's what I think is going on. And I think, you know, especially you can see that when you see the difference in men and women, because, you know, again, in HRS, we're still dealing with a lot of older cohorts where women don't have as much formal education, or aren't participating in the workforce. So I think it's a lot of social things going on across the life course that feed into this. And I think also with the relationship with age is affected by sort of selective mortality, right? So it's who is still alive at older ages. So I think, again, even though we think of this as a biological marker, I think it's a biological marker of a lot of social forces.
Okay, thanks.
Just one more. Childhood IQ. So this is trained in the Lothian birth cohort, and Deary and colleagues showed in the Lothian birth cohort that childhood IQ measured at age 11 using the Mill Hill, and I can't remember what the other one was, correlated about 0.7, 0.8 corrected, I think, with IQ measured when they were 74, 75 years of age. Do you have any administrative linkage to like test scores in HRS or anything like that?
No, unfortunately. I mean, we could. Katrina's working on it. You can talk with her at dinner. The closest thing we are going to have is, you know, some markers of how good people's schools were. So, you know, but not their individual test scores on anything. So we don't have that, which is kind of why some of these types of markers are really interesting, right? You know, because maybe we can get at things that were kind of set earlier in life. Yeah, but Katrina's working on linking our residential history and life history data, including the schools people went to school quality metrics, historical school quality metrics. So it'll be interesting to have that. But yeah, unfortunately, we don't have anything like that.
Walter?
So Jessica, this is Coulter. This isn't really a question for you, but just as a comment. So there are studies like Future Families that are taking it to younger ages and
Yeah.
seeing both the effects of schooling on epigenetic G and also, for example, Future Family, we see it change over time. So we can have two measures of epigenetics and see that it changes, which implies it's not just a pure IQ that's
Yeah.
fixed over time, that there is some ability to modify it, which kind of goes to like, at what point, but even at age 9, their epigenetic G is predictive of if they go to college when they're in their 20s. So it has kind of a long-term predictive capacity too.
Any other comments, questions? No. Okay.
Thank you.
Thank you.
Thanks. Bye.